something down the line. But currently it is a lottery system, and if you haven't gone on one, then your chances increase get on a field trip. So feel free to sign up for one. We also have elections, and pretty easy. There is nothing that's contested, so we are not having to worry about a ballot. It's going to be just us introducing ourselves. So I am going to let Ms. Kim Brown kind of, she was the one who was on our election committee. And so she's going to introduce all of us. If you want to come up. And I can do my part now, so I don't have to come back to the mic. I'm Shayna. I'm going to stay on as president, unless anybody wants to fight me for it. Hopefully, we can have fun next year and get a lot more programs going. I think we've gotten a lot of stuff going so far. And then here is Kim. So firstly, I want to say thank you to everybody who stepped up and volunteered to be on the board, and those. And basically, this is just to introduce everybody, what their position is, and just to say hi. So if you see their face around, don't be afraid to come and say hi and talk to them. My name is Erica Jensen. About midway through the year, there was a vacancy, and I feel like there was some desperation. And they said it was some note taking, which I'm a teacher, so I can do. And yeah, I am the OMS board secretary, and looking forward to another year of doing that. Favorite mushroom? Probably just because I like to say Scutellaria scutellata, the orange eyelash cup fungus. Had to get that one under the microscope before I actually saw the little eyelashes and could really identify it. Odd question. Yeah, you can come up here.
Hi, I'm Ethan. I'm running for treasurer. I heard we needed a treasurer, and I'm an accountant at my job at a publishing company, so I'm stepping up. That's about it. Favorite mushroom. I found for the first time this summer when I was in Michigan, Lactarius indigo. That was pretty fun.
Ready?
Thanks, Ethan.
I'm Alice. I'm the book sales coordinator for OMS, and I've been a member for about 10 years on and off, and just finally decided, hey, just like everybody else, an organization thrives off volunteers and active volunteers, and I really like books, and so that's why I signed up to be sales coordinator, and this is an open board position that I just got. And my favorite mushroom for this year, because I found it for the first time, is the Elfin Saddle, Helpmala vespertina, which is on the table back there, so go check it out.
Oh boy. My name is Kathy McBride, and I'm the volunteer coordinator for all that. And I was recruited by Shayna as I was sitting in the dentist's office, which I have a terrible fear of dentists. And Shayna worked at a dentist that specialized with people who are extremely fearful of dentists. So
that's cool.
Shayna said to me, "Hey, you look like a mushroom person." But indeed, I was starting to develop an interest in mushrooms. And she said, "I need a volunteer coordinator." And my experience in my past work, I no longer work, was really focused on kind of helping organizations realign themselves. And I thought it might be a good fit for post-COVID work, and bringing people back together again. I hope to bring a little bit more process and procedures into onboarding people and using volunteers and acknowledging their work without making it stringent or weird or anything like that, and allowing the creativity of this group to continue to grow. Don't ask me what my favorite mushroom is because I can barely identify anything. So my hope is to go on a field trip real soon and get a lot smarter so I can hang out with you guys and talk.
Thank you so much, and I hope to do some good work with you guys, so I appreciate it. And then next we have our speaker. So our speaker tonight is the 2021 recipient of the Judy Roger Memorial Scholarship. And after talking to her on the phone many, many times, and also, meeting her here, she really does remind me of the spirit of Judy. Fascinated with things, not knowing that she was in love with lichen until just by chance, and then all of a sudden her world opened up. So it's someone with a great sense of humor, and so it's really wonderful that we can offer this scholarship in the memory of Judy. She's originally from South Carolina, and about 10 years ago, she decided with her husband to see what the other coast looked like. And in that spirit of discovery, she just happened upon Arcata and said, "This is wonderful." So at that time, then she decided to do her bachelor's degree in botany at Cal Poly Humboldt. And then she went on to complete her master's degree in biology, studying lichens under the mycologist Dr. Henkel. She now works as a botanist for the Redwood National Park, where she focuses on ecological restoration and rare plant conservation. They do some fungal surveys out there, and she is kind of hoping to get to know some of the mushrooms, but definitely a lichenologist. But she still makes time for fungi, volunteering on the board of the Humboldt Mycological Society and hosting community lichen talks and walks. Please welcome Sarah Conway.
Thank you. Hey, everyone. Thanks so much for having me. And thank you to the Oregon Mycological Society for providing me with part of the funding that I needed for my research. It was very much appreciated. So I have a confession to make actually before we start, which is my research is not actually titled Loving Lichens, as much as I would have liked it to be. Biological master's programs don't typically care about alliteration or like cute terms of phrase. So my real thesis title is a macro lichen inventory of the Horse Mountain Botanical Area. But I wanted people to show up tonight. But I promise we're still gonna talk about fun lichen stuff. I'm gonna give you a little bit of an intro to lichens so that we're all on the same page. And then I do have to talk about my research because that's part of the deal, but I promise not to bog you down too much with statistics and DNA sequencing. So let's get started. I'd first like to thank a few people who helped me along the way. Funding was provided for by the California Institute of Biodiversity, the California Lichen Society, the California Native Plant Society, the Cal Poly Humboldt Biology Graduate Student Association, our Humboldt Mycological Society, you guys, Oregon Mycological Society, and the state of California. I also need to thank my advisor, Dr. Terry Henkel, and the people at Six Rivers National Forest who helped me get all my permitting done. And then this is my dog, my field assistant, Hugin. He deserves recognition for coming out to all of my field work days. And then Horse Mountain, where I conducted my work, is part of the traditional lands of the Hoopaw and the Tsungaway tribes. Can everybody hear me? Okay? Okay, good. So at Humboldt, we talk a lot about the green blur. This is a concept you may or may not already be familiar with. It's basically referring to the perspective held by, unfortunately, the vast majority of the population, where they just fail to recognize the diversity of organisms surrounding them. So they walk outside and they see a blue sky, and then a blur of green where all the plants are, because they can't pick out individual species. It's just kind of green stuff. And then they look at the ground and it's just kind of a bunch of brown stuff. And that's the extent of their outdoor experience. You all have already broken through the green blur. You're here at a Mycological Society meeting. When you go outside, you look at the ground and you don't see a blur. You can pick out individual species. Even if you don't know species yet, you're at least paying attention to some of the mushrooms. And you probably started out by noticing a few charismatic species, and then you fell in love and started noticing them all over the place. And now when you travel, you notice fungi where you are. You start to expect particular fungi to show up in certain habitats, and it just makes everything more interesting wherever you go. It just enriches all of our lives, doesn't it? So I am hoping that through this talk, I'm going to be opening your eyes to a new host of organisms that you might not already be paying attention to, and those are lichens. So I grew up in South Carolina, as Kim said, and I just couldn't believe the first time that I went back home after taking a few mycological courses, and the diversity in my own backyard just blew me away. And I considered myself a pretty observant, like outdoor kid. I was always outside in my backyard, but somehow I was just glazing over all these organisms because my brain didn't know what I was looking at. And so you just kind of, it blurs over. Every class that I took at Humboldt, I felt like I was adding a new lens to my eyes, where suddenly I'd walk outside and I would see this entire host of organisms that I'd been ignoring. So I'm hoping that you all will walk out of here with at least a partial lichen lens after tonight. So why should we bother studying lichens, the scraggly little organisms that you see on tree branches or even sidewalks? In addition to being vital nutrient cyclers, lichens are primary successionists, which means that they are the first organisms to establish themselves on bare land. They are also tools by which we can monitor air quality. In addition, lichens are essential for the survival of some of our favorite charismatic megafauna, right? The animals out there that people actually care about if they're not into mycology. Elk and caribou use lichens for winter forage. Birds and tree voles use them for nest building materials because of their excellent antimicrobial properties. And then flying squirrels in some areas use this lichen here, that's Bryoria, for up to 90% of their winter forage diet. And then who depends upon there being healthy tree voles and flying squirrels to eat, but the beloved and endangered spotted owl? Management practices that affect lichens will secondarily affect these organisms. Lichens also provide camouflage, protection, and even habitat to countless invertebrates like those mites and springtails. And I can't tell you the number of times that I've been looking at a lichen under a dissecting scope and I've been met with a set of eyes just like that. So many cute little jumping spiders. Oh, and then there's, I've got some turret spider photos. Do you know what turret spiders are? They're kind of a California thing. They're native to our tiny area in Northern California, and they build these turrets. And they just build them with any material that they can find. And so I often run into them in the park where they built them using lichens. And you can see that this turret is built using several species of lichen. And I found one more, totally different lichens. On the left, these are camouflaged insects. Well, this isn't an insect, this is a spider here on the lower left-hand side. Can you make out the little legs and the body in the center? And then, do I have a pointer? Can you see? Oh yeah, you can. And then here is the body of the moth here. There are tons of amazing examples of lichen camouflage. I highly encourage you all to Google them, look them up yourselves. But in case you don't, I included a few more. This is a lichen orb weaver. I took this photo right next to campus, and you can see how accurately it's mimicking this lichen right here in color and even shape of the lobes. Pretty incredible. And then I mean that Usnea with the katydid, forget about it. So this is all well and good. They're important, fine. But what even is a lichen? Lichen is short for lichenized fungus. So they are fungi. In fact, they make up roughly 20% of all described fungal species. So lichens deserve a spot in mycological meetings and mycological clubs. I should be having lichen walks down here too. Now, the lichen is similar to other fungi in that they are heterotrophs, right? Do we know that? Lichen or fungi are heterotrophs in that they can't make their own food. They're just like us. Unlike plants, which are autotrophs and can, create carbon from the sun via photosynthesis, fungi have to seek out their carbon elsewhere. They can't make it themselves. So some fungi evolved to be saprotrophs, right, where they are decomposing dead organic matter like leaf litter. Others enter into mycorrhizal associations where a plant enters into symbiosis with a fungus. That fungus is providing the plant with greater access to water and mineral nutrients, and in exchange, the fungus is getting carbon from that plant via photosynthesis. The lichen symbiosis is very much the same as a mycorrhizal association because the lichen is obtaining its carbon via a photosynthesizing organism. So when we think about the lichen symbiosis, it's made up of two main partners, the fungus, which we call the mycobiont. I'm going to have a lot of lichen vocab for you, so brace yourselves. And then the photosynthetic partner is called the photobiont. Now, the mycobiont is an obligate fungus. It cannot live without the photobiont. But the photobiont is going to be a species of green alga or cyanobacteria, and that is free living. So they're just out there doing their own thing. They don't necessarily have to enter into the lichen symbiosis. You find them, they're on the ground, they're on tree bark, fence posts, you name it. You can actually see all these little orange fluffs are a type of green alga. I know they're not green, but that's what they are. And so they're just free living in this state until some cells potentially get captured by the fungus, the mycobiont. Then this changes the entire body plan of the fungus into the lichen thallus, we call it, which is just the lichen body. And so there are roughly 200 species of described photobiont, but there are upwards of 20,000 species of mycobiont. So there's a lot of overlap whereby one photobiont might enter into symbiosis with hundreds of different mycobionts. So when I talk about a species of lichen, it would be really taxonomically confusing to try to name them based on the photobiont. So a lichen species is based on the mycobiont. Oh, and then here I'll show you, this is a bigger picture up at the top where you can see this white fluffy layer is actually just fungal hyphae, right? And then this green layer here is the photobiont, that's the green alga layer that the fungus is containing. So that fungus is able to provide that alga with a little extra shelter and water and mineral nutrients, and then that alga is providing the fungus with carbon because it's still able to photosynthesize. This is a really cool SEM scanning electron microscope photo of a really common lichen, Parmelia sulcata. And I didn't take these, this was done by a group called the Eye of Science. And you can see here this layer of the photobionts and the hyphae that are just surrounding them physically, and they form that network of nutrient exchange. So the fungus gets the carbon, and the alga is getting some water and mineral nutrients that the fungus can just absorb from its surroundings. So a little more lichen vocab for you. This top layer is called the cortex. All of these terms you kind of need to know if you're going to start identifying lichens. So I'm sorry, it's a little vocab heavy, but if you are going to start, I recommend writing them down or just kind of saving them in the back of your brain for when you get a lichen book. So this is the algal layer, that photobiont layer, where the nutrient exchange is happening. And then this is the cottony medulla. It's basically just loosely woven hyphae. And actually, if you go out and rip open a lobe of lichen, you'll probably be able to see, just with your naked eye, that little cottony medulla. And then some lichens also have a lower cortex, which is just the lower skin. And this we call conglutinated hyphae. It's the same thing that happens on like this, the cap of a mushroom, where you can peel a little bit off. It's all stuck together. Same concept, just tightly woven. I have to also mention that this is the simplest definition of a lichen. We hardly know anything about them relative to other groups of organisms. We don't understand fully all the components that make up a lichen thallus. We've yet to be able to grow a lichen in vitro in the lab because we don't know what all the components are. Yes, there is a primary fungus, the mycobiont. Yes, there is some kind of photobiont, but we're discovering that there's potentially additional fungi in the thallus and basidiomycete yeasts. And then we also think that the microbiome is potentially playing a role in the thallus assemblage. So there are a lot of unanswered questions out there for any of you budding lichenologists. More lichen vocabulary, but these are main body plans of the lichens that you really need to know if you're going to ID them. On the top right, this is actually that lichen that they did the SEM photos of. That's Parmelia sulcata. So you can just picture that view was into one of the tips of those little lobes there in the top right. This lichen on the left is called a fruticose lichen. They are pendant, shrubby, kind of branched. Some of them are much more pendant than others, like this Ramalina, and others are more erect and shrub-like. The Parmelia on the top right is foliose, so there's a distinct upper and lower surface. I kind of remember that like foliage, like a leaf. And then one of my favorite words, squamulose, for all of these tiny little overlapping lobes that you see here at the base of these cladonia cups. And then finally, there are crustose lichens. I've been seeing them all over the sidewalks in Portland the past couple of days. And crustose lichens are flat against their substratum. So, and many of them actually are embedded in the substratum, whether that's rock or a tree branch. They don't have that lower cortex and they're directly embedded. When that's the case, when they are embedded, they're referred to as microlichens. Now, you might have noticed that my thesis is a macrolichen inventory. That's not because I don't love the microlichens, it's just because honestly, the expense and time required for identifying microlichens is far beyond the scope of a master's thesis for the size of the area that I was trying to survey. So I just stuck to the macrolichens. But that's not to say, here's an example of those microlichens embedded in that rock. And then this is a macrolichen up here. It's got an upper and lower cortex. I can just peel it off of whatever substratum it's on. This is one of my macrolichen specimens. So it's not like all macrolichens are large and microlichens are small. It's all about that embedding. Plant people in here, if there are any, might recognize this as the tip of Caliceidris decurrens. It's the California incense cedar. Tiny little scales. So this is actually only a few, the entire thallus, this entire brown thallus here, is only a few millimeters wide. But I could peel it right off of those scales. It wasn't embedded. Therefore, it was a macrolichen and it was fair game for my inventory. So we know that they are tiny and they are complex, but we also know now that they have valuable ecosystem functions and are essential for preserving biodiversity. When you don't have data on a host of organisms, you have no way to help manage or protect them. And that brings us to my actual thesis, which is a macrolichen inventory of the Horse Mountain Botanical Area in Six Rivers National Forest. It's the first inventory in the coast ranges of Northern California, which is thought to be one of the highest diverse locations of macrolichens on the West Coast. So why Horse Mountain Botanical Area? you can see that it is located just west of Willow Creek. It's about 1,100 acres in size. And in this map that I borrowed from Backcountry Press of the serpentine outcrops in Northwest California, you can see that the Horse Mountain Botanical Area is sitting directly on top of one of them. Now, serpentine is a type of ultramafic rock that's high in iron and magnesium. So plants that can exist and have learned to evolve to adapt to these soil types are typically really unusual themselves, and often they're rare. There's also a diverse assemblage of conifers in the botanical area. One you all may be familiar with, this is Chamaecyparis lawsoniana, the Port Orford cedar. And the Port Orford cedar reaches the southern extent of its entire range on Horse Mountain. There's also a population of Jeffrey pines that occur unusually close to the coast for the species. While a lot of lichens are epiphytes, meaning they grow on other plants and trees, that means that if I have a diverse assemblage of conifers, it lends itself to a diverse assemblage of macrolichens, which is what we were hoping for. Oh, there's epiphyte, grows on trees. So here were my objectives. I set out to survey the Horse Mountain Botanical Area for as many lichen species as possible. I then wanted to identify all of those specimens to species and then curate extended voucher specimens, which I'll explain. And I wanted those extended voucher specimens to contain molecular data. So I set out to obtain ITS barcodes from as many species as possible. We'll talk about that too. And these three combined to be my primary objectives, which is really just to set a baseline of data on lichens in the botanical area. And finally, I decided that I wanted to characterize the macrolichen community up there and compare it to plots from other nearby forests to see how the diversity and composition of Horse Mountain stacked up around the surrounding areas. So I first had to survey the botanical area for all these lichens, but how on earth do you survey 1,100 acres for lichens when most branches look something like this? Luckily for me, the Forest Service already has a protocol in place where you select just 1 acre circular plots, and their protocol dictates that you only need 1 of these plots per square mile. So for me, I would have needed 2 plots for the Horse Mountain Botanical Area. But I really wanted to be thorough, so I did 20 plots. And I tried to cover as much of the botanical area as possible. You'll see that I ignored a couple of areas. This used to be an old copper and nickel mine back in the early 60s. It was shut down in the early 60s, I should say. And then this area here, there's some communication towers up there. And basically, I'm just not interested in the anthropogenic effects on macrolichens for now. So I tried to spread these out between, I tried to cover the entire range of elevation. So I hiked way down into some of these valleys. It goes from about 1,200 meters to 1,400 meters. And I had to do a lot of recon and hiking around the botanical area before I even chose my plots, because I couldn't just randomly choose them. Similar with mushroom studies, you have to look for these specific habitat niches. And if I were to miss one of those niches, I can miss a huge hotspot of biodiversity. So I really had to be thorough in combing over the entire botanical area before choosing these locations. So for each one of these 20 plots, I followed that FIA protocol that I mentioned. It's called the Forest Inventory and Analysis Protocol. And they do this for many different organisms in addition to lichens. So like I said, there's a 1 acre circular plot. You don't have to worry about the little subplots. I would just use those to kind of orient myself within the plot. And I'd walk through in a sinuating fashion that would allow me to cover the entire plot. It would kind of look something like this. And I've got my hand lens out and I'm inspecting trees and branches. And there's so many aspects of this protocol that I don't want to bore you with tonight, but essentially I go around and that's me, and I'm looking at lichens that are epiphytic. The Forest Service protocol dictates that it can only be epiphyte, so only lichens on trees. So I'd start from a certain height above the ground and go to as far as I can reach. So I really just have this little narrow band of focus. And I would go through the entire plot just looking at that narrow band, collecting as many species or morphospecies if I didn't recognize them that I saw. I'd put them in these little paper packets. I'd write down the substratum. I'd write down the tree species that they were on. I'd give them an abundance rating, which is like a logarithmic scale dictated by the Forest Service too. There's Hugin keeping watch over my specimens. And this is that little abundance scale. It's pretty straightforward. You just assign a number 1 through 4 based on how many individuals you're seeing. But remember, I wanted to survey for all of the macrolichens in Horse Mountain, not just this little narrow band of epiphytes. So I had to figure out a way to include all the lichens, but I also wanted to maintain this protocol in its exact form because I want to be able to compare my data to the thousands of FIA plots across North America. So what I ended up doing was adding a separate survey to each one of my plots, and that was my ground survey. So I would look at everything from, this is me going around with the epiphytes, and then I would look at everything from below where I left off with the epiphytes, and this would include down tree branches, stumps, rocks, the soil itself, theoretically covering everything except the high canopy. And I'm hoping that because there's enough windfall and fungal disease centers up there, that there are enough trees down on the ground where I was getting a pretty good look at the canopies themselves too. So I would do 2 surveys for each one of those 20 plots. And these are my little hand lenses that I've got to carry around, and I'm looking as I'm walking, and you're probably thinking, like, what could you possibly be looking at when you're trying to go that quickly? And I didn't mention I have a time limit of 2 hours for each one of these surveys, so an entire acre in 2 hours. But if you've ever done botanical surveys or fungal surveys, you kind of know that it gets really quick when you're walking through. You can start to pick them out. Your brain's really good at recognizing different patterns and textures. So as long as you're focused, it's actually quicker than you would think. So what would I be looking for with the hand lenses? More lichen vocab. These structures, mostly. On the left, these are apothecia, apothecium, singular. And those are the sexual reproductive structures of a lichen. Those are the spore-bearing structures. And actually, they're ascocarps, cup fungi, same concept. And this top layer, that surface layer of the apothecia, are actually really similar to the gill surface of a mushroom. They're giving off spores. Those little spores are going to germinate somewhere, and they're going to hopefully find a little photobiont to capture so that they can become their own new lichen. So those are the sexual reproductive structures. These are both asexual reproductive structures, and you have to know them if you're going to identify lichens, because certain species just have particular forms of these. The Cerridea in the center, they really just look like powder, like teeny grains of sand. And that's because they're just little bundles of hyphae surrounding individual cells of the photobiont. So they're the whole symbiosis package. They're going to blow off of the lichen, hopefully land somewhere habitable for them, and they will develop a cortex, develop that skin, and grow into a full lichen. That's actually a clone of the parent lichen. And the same thing with the Isidia. Isidia are just a little more advanced than Cerridea. Same concept. It's the whole symbiosis package. It's hyphae wrapped around individual photobiont cells, and they already have a cortex. So they're just a little more weatherproof. They can stand up to a little bit more difficult conditions. And my lichen professor, Marie Antoine, told me when looking for them, because sometimes it's hard to distinguish the Isidia from the Cerridea, she said, look for tiny, shiny sausages. And it's actually really accurate. They're tiny, shiny sausages. That's it. Here are some of my fieldwork photos. There's a lot of Pinus lambertiana up there, and Hugin would always bring me the cones. This is a gorgeous Cladonia specimen that has stacked podetia or fairy cups. And then up here is what we call a jelly lichen. It's a gelatinous lichen. And these are non-stratified, so they don't have that same pattern that I showed you. They're basically just one jelly unit. And this one is actually wet, so you can see it, but when they're dry, they just look like crusty soil, essentially. But you develop an eye for those too over time. And then there I am with my rock chisel and hammer, and that's what I would use to get my rock lichen specimens. Just to give you an idea of what I was looking at out there, it's funny you say that. Most people, when they start to learn lichens, Usnea is a really great intro genus because it's got a characteristic about it that is foolproof. Have you seen that, the little stretchy rubber band in the middle? Yes, we love Usnea for that. But this is not Usnea. So you gotta really pay attention with lichens. You gotta get up close, you gotta have your hand lens. This is Alectoria. Almost all of this tree is covered in Alectoria. And most Alectoria species that I was dealing with don't have very many apothecia, those little spore-bearing structures. They're pretty pendant, they're not very shrubby. But if you're out there with your hand lens and you're cruising through and you're paying attention, you might notice this gorgeous species, which is Alectoria lata, with its little moon-like apothecia. It's much more shrubby, has way more apothecia. And based on my molecular data, it might not even be an accurate name for this area, but that's a story for another time. This is a really common lichen that you run into. It's called Platysmacia glauca. It's called the tattered rag lichen. We need more lichen common names. Even though I'm not a huge common name person, I feel like it gets the public involved. And so few lichens actually have common names, but this one does. And it's fitting. It's pretty tattered. The edges are a little eroded. The bottom is kind of mottled and brown. And so you'll see a ton of this. But again, if you've got your hand lens out and you're paying attention, you'll find this fun species, much less common. That's Platysmacia wheeleri. And you can see that the only distinguishing mark here are these undulating margins that are covered in that powdery ceridia. This is one of my favorites. This is kind of a treasure hunt lichen, Seroma hypnorum. And you can see how large the moss is in the photo. These apothecia are probably only 2 millimeters wide. They're really tiny. But I really like the pearled projections around the apothecia, very elegant. This is a cool species, Umbilicaria angulata, these neat angular apothecia. And this is another example of, if you're not paying attention, you're just gonna miss the species entirely, because it looks almost identical to a super common lichen, Umbilicaria fea, but Umbilicaria fea has round apothecia. Oh, and all Umbilicaria have these cool folds, which is just to increase the surface area for the spore bearing, which is exactly the same as gills on a mushroom. And here is that exact same specimen just in my hand, that you can see that you need to be paying attention if you're going to see that some of those apothecia are angular and not round when you're walking by a rock that's just covered in this species. And then this is the poster child of lichens, Lotharia columbiana, with its little, the one on the left here, that's a hypogymnia on the right, don't worry about that. The little sunshine projections around the apothecia. And it actually kind of has a dark history. I don't know if you're familiar, but Lotharia, like most lichens, produces secondary chemical compounds that are unique to lichens. And Lotharia species produce something called vulpinic acid, which is toxic. And back in the day, homesteaders and ranchers would feed Lotharia to wolves and foxes that were messing with their farm animals. So they'd stick a clump of Lotharia in a piece of meat, feed it to the wolves, and inevitably kill them. But now we get to look at them and they still look sunshiny, and people don't use them that way anymore. So I surveyed the entire botanical area, and I ended up with just under 900 lichen specimens, 888 to be exact. So it was now time to go to the lab and identify all of these specimens and then curate extended voucher specimens for each one of the species that I encountered. So identifying lichens can be super quick. You can site ID them, like this Usnea intermedia. It's one of the only Usneas that has apothecia in our area, so you can just pinpoint it. You don't even have to use a hand lens. You're like, great, I know the species, boom, done. Other times, not so easy. You need to start with some microscopy. I use both a dissecting scope and a compound scope, depending on whether I'm looking for micro features or macro features. And often I would just use my dissecting scope to make cross sections of the apothecia because they're so tiny, and I need to look at them under the compound scope to look at their spores. So here's, just for size, there's my finger and a little apothecium that I've sliced into, preparing it for the compound scope. Here's what that looks like just under the dissecting scope. And then when you get it under a compound scope, it might look like something like this. And this is one of my specimens, and you can see here is an ascospore. You might already be familiar with those if you're doing some fungi microscopy. And I would look at the size and shape, and then I'd try to count the number of ascospores per ascus, which is this kind of case. You can see them all jumbled in this like long sac. That's the ascus. And you can even see a little algal cell down here. So that's what I'd be looking for if I was trying to ID something with the compound microscope. And sometimes it wasn't enough. Sometimes they're too morphologically similar and you need chemical spot testing to get to the bottom of it. This is just the same as you all walking around the forest with a little bottle of KOH. You're dripping KOH on the mushroom cap. You'll get a color change, hopefully, and that will help you determine the ID. Same concept. We use a variety of chemicals, and the most common is the C test, which is just bleach. And then there's the K test, which is 10% potassium hydroxide. And the P test, which is p-phenylene diamine crystals dissolved in ethanol. A little bit harder to come by. And if the chemical spot testing was successful, you would end up with a nice little color change, like I have in this Cladonia. Turn bright red. Great, now I know the species. Sometimes even chemical spot testing was not enough, and you have to resort to molecular work, aka DNA sequencing, which we will talk about. So this is kind of my command center, if you will. I spent so much time at this lab bench over those microscopes. And there's another example of a little spot test that I was doing. And you might wonder why I included that semi-blurry photo, but I just wanted to show you the scale of some of my spot tests. All of this is happening under the microscope. So let's start building these results. Of the 888 specimens that I collected, I was able to identify 865 of them to species. 14, thank you. 14 of those were microlichens that I ignored, and 9 of them, 9 tortuous specimens, I could not ID to species. Most of them were like too fragmented, and some of them were just too tiny for me to be able to identify, but there are 9 that I just didn't get to. But of the 865, I was able to find 119 species within 52 different genera and 17 families. And when Dr. Bruce McCune and I set out to look at this research, he's a lichenologist up at OSU, we were expecting roughly 60 species total for the botanical area. So this was super exciting, almost double what we expected. And I ended up finding between 20 and 44 species per plot with an average of 35. Now, even though I found more species than I expected to, I still couldn't be sure that I had successfully, thoroughly surveyed the botanical area until I built one of these, which is just a species area curve. You can see on the x-axis, that's my number of plots all the way up to 20, and then the average number of species. So as you survey more and more plots, you find more and more new species and your curve continues to grow. But ideally, if you're finding everything, then you're going to encounter fewer and fewer new species over time, and that line is going to start to plateau. My line is starting to plateau around the plot 15 mark, and I gained less than 10 species in the final 5 plots that I did. Jackknife estimates gave me an estimated 160 species for the botanical area. So that means if I were to survey infinitely, just forever and ever, I might get 160. And then a bias corrected form put that at 143. So with 119, we felt confident that I had thoroughly surveyed the botanical area and we could move on. So let me introduce you to the new Cal Poly Humboldt Lichenarium. This is actually a photo of my lovely lichen professor's desk. The Lichenarium is on the adjacent wall, but it's just cabinet, so I thought this was prettier. But that is where they're going to house all of my little packets, and then we're also going to store thousands of local lichen samples that we have from other studies that just haven't had a place. They've just been sitting in cabinets at the school. So we are now digitizing them, and that brings me to talking about the extended voucher that I was discussing earlier. So it's extended because it's not just a paper packet in a cabinet. And those paper packets and cabinets are still useful. People can mail them to each other to do individual studies, research, but it's much more valuable to have those specimens digitized with photos and additional metadata so that people from all over the world can use them without having to physically look at the specimen. So I created a bunch of extended voucher specimens and then digitized them all to CLH, which is the Consortium of Lichen Herbaria. Once you all become obsessed with lichens, you will use this website quite often. It's an amazing resource. You can look up lichen photos. You can see lichens by checklist, by species. They have them geo-referenced. It's really incredible. And I'm proud to be contributing even in this tiny way. So for my extended voucher specimens, I curated 287 of them, and I tried to do 3 voucher specimens for every species, just in case I was wrong with my IDs, just to make sure that, you know, if somebody comes along 10 years from now and has a question about one of my species, they have a couple of other packets that they can compare to. 3 vouchers per taxon, and then 34 of these taxa species were new to CLH for Humboldt County, which is cool, but also kind of surprising because these were commonly occurring species. They were not super rare. It's just going to show how little publicly available lichen data is out there. So yeah, exciting for me, but also kind of depressing. We need more stuff out there. So first 2 objectives completed. Now I wanted to add that metadata. I wanted those extended voucher specimens to have the DNA sequencing. So I set out to get those ITS barcodes. Some of you may already be familiar with DNA barcoding, very popular with fungi, and ITS is a great barcode for delineating species of fungi. Essentially, for people who aren't familiar, instead of having to sequence the entire genome of an organism, which is incredibly expensive and potentially impossible, you can just look at a tiny section, a little barcode that is going to help you delineate species. And the way that I did that was by looking at this particular barcode, which is used well for fungi and lichens, which are fungi. This is the ITS, or internal transcribed spacer region. Don't be scared, we don't have to talk about all of this. I'm basically just showing you that the ITS region in eukaryotes, so plants, animals, fungi, are both of these little regions, and they're non-coding. And that's the whole thing about them that makes them great for delineating species, because these are structural ribosomes on the other side, but this little section, it's not doing anything. It's just a spacer. It's not coding for proteins. It basically has no information in it, which means that there's not very much evolutionary pressure acting on it. So there can be a huge amount of variability between really closely related species, which is great for us. So I would use these little primers, and I use ITS1F and ITS4 for those of you who do this. And basically these primers can locate the little region that I'm looking for, and then I would amplify that region, make a bazillion copies of it, so that when I sent my little DNA sample off for sequencing by these big DNA sequencing machines, those machines can see just this region because I've copied it so many times, it's not bogged down by the rest of the data in the genome. And the way that I would amplify or copy these regions is through PCR, which is polymerase chain reaction work. And anyone in here who has done PCR knows that it is a lot of pipetting, a lot of gel electrophoresis, and a lot of wishful thinking. I spent so much time doing DNA sequencing. I attempted every single species, and I only ended up with a little more than half being successful. And I did multiple attempts on the species that weren't working. So it was a lot of sequencing. But but it's extremely valuable for lichenologists all over the world who are doing taxonomy. They don't just have to be in this area, because if they're looking at phylogenies or like lichen family trees, this is the data that they need to put all that together. So when I would get my sequences back from those big sequencing machines, I would analyze it, annotate it, trim it up, and then make it pretty so that I could deposit it into GenBank, which is part of the National Center of Biotechnology Information. And that way it can be accessed from anywhere around the world. So I ended up with 81 good quality ITS sequences from 63 species, still technically the majority of my species, if only by a little bit. And 9 of those species were brand new to GenBank, which again, cool, but if you compare that to some of the commonly found mushrooms, there are hundreds of ITS entries on GenBank for each one of those species. So we need more people doing ITS work on lichens. And you might be wondering, how do you do that for lichens when they are a complex symbiosis? Actually, the primers are so specific that I don't even have to extract hyphal structures or just isolate the fungi in any way. I can just take a tiny piece of a thallus, like 1 square millimeter, drop it into my little chemical solution with the primers, and the primers can find that spot. They just don't even pay attention to the alga, which again, great for me. Through this molecular work, I was able to describe a brand new species of lichen, which is very exciting. This is Hypogymnia tuckerae, and Bruce McCune helped me get this published in the Bryologist. And we named it after Shirley Tucker, who's a female lichenologist doing incredible work for lichenaria these days. And I actually found 3 specimens up on Horse Mountain. This is one of them, and this is now the type specimen for the species. And the Forest Service is trying to make it a Forest Service sensitive species, get it listed, which is cool because then it's going to be included in surveys and protected in project areas in the future if it does get listed. And I'll be really interested to see where it turns up. All right, primary objectives finished. Let's look at this macrolichen community as a whole and compare it to surrounding forests. This is where it gets a little stats heavy, but I'm going to breeze through it as much as I can. I use non-metric multi-dimensional scaling, which is NMS, to create ordinations. I basically built a bunch of matrices with my metadata from my plots that look like this. It was just like a billion Excel spreadsheets that end up getting multiplied together in a software that turn them into this little picture, which basically just shows how different my plots are in species space, meaning like this plot has a different group of species than this plot that's much farther away from it. The closer they are, the more similar the plots are. And again, I'm not going to get into the nitty-gritty here, but there are a bunch of plot attributes that I measured to kind of beef up my extended voucher data. So I measured slope in every plot, aspect, elevation, distance from the plot center to the nearest stream, total basal area of all the trees, and then the proportion of the basal area that was made up of conifers versus hardwoods. And it ended up being that the latter 3 attributes were actually significant in the macrolichen community composition. And an easy thing that you can actually see here, I'm pretty sure, is that these three plots have a totally different composition than these over here on the left. They're super far apart. And you can see that conifer vector pointing this way. This means essentially that there were only conifers in this yellow circle, whereas there were majority hardwoods in the orange circle. And we've known for a long time that particular lichen species have a preference for hardwoods over conifers, but it's sometimes just really nice to see that reflected in the actual data. So I also included functional traits to see if maybe the functional traits had an effect on that macrolichen distribution. And functional traits are just those that are part of the ecology. So that what is that lichen contributing to the ecology of an area? For example, these are all the ones that I use, which were the ability to fix nitrogen, as is the case for what we call cyanolichens. So cyanolichens have cyanobacterium as their photobiont. It's not green alga. And if you know anything about cyanobacterium, it fixes nitrogen. Just like in the nodules of bean plants, they use cover crops, same exact concept. I also looked at forage value for wildlife, and that's essentially just the pendulous lichens that are accessible. And then obligation to rocky substrata, we call those saxicolous lichens. And then affinity for nitrogen-rich environments, and these are nitrophiles. And I would think, rocky outcrops where birds like to defecate, or areas that are near agricultural lands. So 100% of my plots contain cyanolichens. And actually, some of the plots contain as much as 30% of the diversity in cyanolichens, which is huge. Nitrogen fixation is really important. 100% of my plots contained pendulous lichens that are important for forage value. 65% contain saxicolous, and only 35% contain nitrophiles, which goes to show that the Horse Mountain Botanical Area likely is an area of low nitrogen deposition. So then this is why I wanted to keep those plots separate, right? My epiphyte survey and then the ground survey, because I wanted to do this comparison. So I took my 20 epiphyte surveys and I compared them to 20 of the surrounding national forests in Siskiyou County, Humboldt County where we were, and also Trinity County to see how Horse Mountain stacked up. So I ended up with 106 epiphytic species total between all 40 plots. 59 of them were shared species between both sets. And then 28 were found in FIA plots, but not on Horse Mountain. 19 were found on Horse Mountain, not in FIA plots. And at first glance, you might think, oh, that means Horse Mountain Botanical Area isn't quite as diverse as the other plots. But remember, all of my plots are in one concentrated area and one little forest type, whereas the surrounding FIA plots are a huge spread of points in a bunch of different forest types. So when I actually made box plots of the richness, you end up with significantly higher richness in the Horse Mountain Botanical Area. Oh, there we go. My median richness was 24, whereas FIA plots was 19. And you can see that my little whisker here extends even beyond their outliers. So the Horse Mountain Botanical Area is more diverse than the surrounding areas when it comes to macrolichens, which is what we predicted. If there are any lichen nerds out here, this is just a list of the dominance curve results. So this just means that these are the 10 most prevalent species in both sets of plots. Orange is showing you ones that are similar, white is dissimilar. And so you can see that the top 5 are pretty similar, but once we get into the lower 5, not so similar at all. It's really fascinating. I really do think that that serpentine influence is having a large effect. So in the future, I really hope that students at Cal Poly are going to continue surveying my plots. We have a lichenology course, and I think it would be a great way to just get them some practice out in the field and continue to see how this macrolichen community changes over time. If they wanted to, someone could add rock type analysis to my study. You could see what lichens preferred, what type of rock chemistry. You could add canopy surveys if you love tree climbing. That would be super valuable. Or if you're really into DNA sequencing, you could try to sequence the photobionts from all my specimens. You wouldn't even have to do any field work. I already have them all collected for you. I'm also really interested to see what the macrolichen community composition is going to do if the area burns. And it's actually come really close to burning the past 2 years in a row. So this is the botanical area there, orange, and this is the lightning complex fires from last year. And they actually had to run a dozer line through the lower part of the botanical area to protect it from these fires. So I talked to a lot of fire experts in the area, and they're like, it's inevitable that it's going to burn. When it does burn, we're going to have a perfect natural experiment ready to go. We've got the baseline data. Now, I won't be that sad if it burns. We'll just get to see what happens to the community in the future. This is a photo of me, not of me, but I took it while standing up on Horse Mountain. You can see the fire in the background, a few ridges over. And I hope that all of my data are used by lichenologists around the world to influence their own studies and find phylogenies. And I hope that you all have learned a little something along the way. I really hope that if you go out and start your own lichen journey, you get this book for this area. It's the one to get, in my opinion. This is Macrolichens of the Pacific Northwest. Y'all should add that to your little book array, OMS. This is by Bruce McCune and Linda Geiser. It's got an awesome intro that talks about all the things I discussed in the beginning, goes into way more detail. We've got color photos of all the species that you're most likely going to run into. And they've got descriptions, ecology descriptions, chemical spot test results. It's an amazing resource. And an illustrated glossary. Hand lenses, critical. You have to have them. These are my favorites. They're Bosch and Lohm, but you can get cheaper ones. I've seen them for like $10. You can get away with a magnification of like 10x. I have a 14x and a 20x, but I think 10x is fine. And I also tell people that collecting lichens is not quite like collecting fungi. Just always have to remind people of that, because I want people out there collecting lichens and studying them, but we know how important they are now. And you can't just remove a ton of lichens from a system and expect everything to be okay. That's why I do love eco-dyeing, and I think it's so cool to dye with mushrooms. But when you pick a mushroom, it's just like you're picking an apple off of a tree, right? The mycelium is still intact, you're not hurting anything. But when you collect a lichen, that's the whole lichen. That's its entire existence. Some lichens take hundreds of years to grow. They're really slow growing. So you might pick up a thallus that's this big, but it's been around for 200 years. So I just tell people to proceed with caution, make sure that you're not, over collecting. There's no reason for you to collect off of substrates either. You don't have to rip a lichen off of a tree. You can probably just ID it while it's on the tree, or look at the ground. There's so much lichen litter all over the ground. Anything that's on the ground actually isn't going to survive because they get too waterlogged. They can't go through all their cellular processes, so they're not going to make it. So that's all fair game. Go for all the lichens on the ground. If they've broken off of rocks, go for it. But don't take too much at the same time because, again, nitrogen cyclers. And if you run into any questions in your inevitable future lichen obsession, please just feel free to email me. I'm at humbotany@gmail. I have an Instagram that's also humbotany. And then these are two other lichen Instagrams that you might like: California Lichens, California Lichen Society, and Northwest Lichenologist up here in y'all's neck of the woods. They always do forays or workshops, all kinds of lichen opportunities out there. So thank you very much for having me. Questions? Questions are the scariest part, so it doesn't matter if there aren't any. Leah, I recognize you from Instagram too. Hi.
So first of all, I just want to say thank you for a great presentation. We have a lot of things that are scholarship people, and they're not always good at making it so that people who are not already familiar with their thing can understand it, and you've done an excellent job.
Thanks. I hope I didn't bog y'all down with too much. Mm-hmm.
I know that in somewhat recent years, there has often been found to be an additional fungus in the lichen.
Totally.
Is that ever an issue?
It is an issue, actually. I didn't actually run into that, but you can totally pick up other fungi, and I guess I did run into that. I just didn't run into those Basidiomycete yeast that they think are present in some cortexes. But I definitely ran into some fungi where it just on GenBank, it would match some tiny little uncultured fungus, giving me the clue that there's something else going on in there. And that would just be something that I have to redo that sequence and hope that I got the primary mycobiont. But yeah, good question. And there was a question over here too. Mm-hmm. Yeah, good question. So lichens are pretty amazing in that you don't have to freeze them. As long as you just dry them out entirely, they can last for hundreds of years because they actually just go dormant. Their cellular processes, when they desiccate, when they dry out completely, no more cellular processes are happening. So until you reinvigorate them with water, they can just sit there. And I've had people reinvigorate lichens that have been in herbaria for hundreds of years in Europe, and they were still able to get successful DNA sequences out of lichens that had been in a packet for over 100 years. Super cool. Anything else?
If they're keeping the cyanobacteria or the algae, and that's what's creating food, what does the substrate, as far as like nutrients?
Awesome question. It doesn't. That's why you can see lichens just like on a metal fence post or on the sidewalk. They're just these little rhizines, but they're just root-like structures, but they're not root-like in that they're absorbing anything. They literally are just for attachment. So you can see lichen growing on your car if you leave it there for too long, or lichen on a window. There's just, yeah, it's getting nothing from its substratum. Really cool. Leah again.
Follow-up question to that, is there are certain lichens that will only grow on crops or only on trees?
That's true. They're influenced by the pH of their substratums, just because they're less tolerant. It's not that they're uptaking any nutrients from it, but they might be adverse to a high pH of a conifer bark like redwoods. You hardly see any lichens on the redwoods themselves. There are only a few species that really like to grow on them. So as I work a lot in the redwood forest, it's kind of low diversity for lichens, whereas an oak, which has, you know, much lower bark pH, and it's going to have all those extra cracks and crevices, the particular lichen species are going to go for that. Good question. Never. Impossible. Yes.
So the microbiome, you're saying, whatever word you use, invade or
capture, we say.
So in that picture you had,
Mm-hmm.
could then a microbiome, microbiome colonize that?
It probably wouldn't colonize like a large piece of that photobiont, but if there are little propagules of that photobiont that are just starting out and they're only a small cluster of cells, because if you think about the mycobiont when it first germinates from that spore, it's also super, super tiny, and it's not going to be able to grow very much until it has a mycobiont, which actually, side note, super cool, sometimes lichens can attach to the wrong photobiont, actually one that they don't really want, just to kind of like be a substitute to get them along. And then when they find the real mycobiont that they want, they'll kill the first one and take the one that they actually are desiring. Pretty cool. Yeah, good question. Yeah.
As far as there's a lot of times you'll see instead of dying trees, have a lot more.
I'm so happy you asked this question.
Are they contributing?
No. Yes, I love this question because that's such a common observation. And even if you don't know anything about lichens, you see dead tree covered in lichens. Oh my God, the lichens killed the tree. You'll actually see there's a huge market in agriculture for pesticides specifically used to kill, I guess they're herbicides, I guess, kill lichens on fruit trees. And I tell people, no, that's helping your fruit tree. The extra nutrient cycling, you want lichens on your tree. So when you see a dead tree covered in lichens, it happened the opposite way. The tree died first, and the lichens were like, hey, look at all this space we have to colonize. It's perfect for us. There's lots of light. It's nice and dry up here. Yeah, I'm so happy you asked that.
Like on an apple tree, covered in
No, that's a healthy apple tree. Yeah. And it's not just nitrogen. I keep saying nitrogen, but there are tons of, the lichens are just absorbing mineral nutrients out of the air. So from rain, from fog deposition, I could talk about this for so long, but there are so many mineral nutrients that then are just, when it rains, they're dripping off of the lichen thalli just into the soil around your fruit tree, adding to the mineral nutrients below the tree. So keep all your lichens. You want them. There are. I'm glad you asked that too. So I have a tea that I really like that my friend brought me from Iceland, and it says that it's moss tea, but I looked at the ingredients and it was a lichen species that they use, Citraria. Common misconception. And so I really like, it's kind of bitter, but that's a species that you can use. I was talking to someone earlier about Usnea. Usnea is getting a lot of rap lately because it is anti-tumor, it's antimicrobial, it has all these cool properties. And so people are using them in broths and medicinally. And I just say, that's great. And Usnea is one that you can actually ID properly, but proceed with caution because too much of a good thing is toxic when it comes to lichens. So I would say if you're doing a pot of broth and you want to add some Usnea for the health benefits, you're talking about like a small handful. You don't ever need more than a thallus or two to add to anything. I would be very cautious. And then there are some people who say that you can eat lobaria, which is the big flat lichen. They call it frog pelt lichen out here. I think that you would have to be starving and very desperate. I don't think it's good, but if you were maybe stuck in the forest, yeah, I think you could get by with some of that for a while. But yeah, overall, I just caution people, and for the same reason I caution people against eco-dyeing, unless you know the species that you're looking at, you don't know what kind of secondary chemical compounds on there, you don't want to eat some Lotharia and end up like the wolves. So caution. Also, side note on the same category, I have been seeing this a lot lately and it's a pet peeve of mine, so I might as well just tell you all. The misconception between moss and lichen, you see these bags of moss at craft stores. They're green, that you want to use them in your pots or your wreaths or whatever. It's super cute, I totally agree. Most of that is cladonia. It is illegally harvested from the Arctic tundra, where it is the main source of food for caribou. It's extremely slow growing. Everyone that I've talked to is like, oh no, I get it ethically sourced. And like, it's literally impossible for you to get it ethically sourced because it's so slow growing. So when you're at those craft stores, beware. It's just cladonia that's dyed green. So sad. Tisk tisk.
Lichens can grow really slow for a long time. Do you have any sense of how old the lichens were?
I don't, because we don't really have great data on most of those species. There are only a few where we kind of have an idea like, okay, this lichen might branch once every year. So then I'm looking at an Avernia species and it's branched 50 times. I'm like, oh, this might be 50 years old. And then there are some claims, and I haven't read any actual papers about this. I don't know if this is true, but it wouldn't surprise me that, you know, map lichen, it's like a bright yellow lichen on rocks. They say that some of them are 5,000 years old. Again, cannot confirm, but I've heard that from reliable sources. We've also sent lichens into space. They're super hardy.
So the lichen used to kill the wolves. What is the toxin in that?
It's called volpinic acid.
And how did it kill them?
I don't know the details of how it gets into their biology. I just know that it's a toxic chemical only produced by the lichen. Yeah, I wish I knew more about the chemistry, but I don't.
Have we seen any environments that have had like a detrimental effect to lichen and environment, what effect that's had in that environment?
Yeah, it's kind of cool that you asked that actually. And I wish I had boned up on this paper before I came here tonight, because there was a really cool study that I learned about in one of my lichen classes based in Portland, where a lichenologist started noticing in this new neighborhood that she moved to that there was kind of a low lichen diversity. Just on the sidewalks and the neighborhood trees, she was just noticing that they were kind of lacking. She decides to make this an actual study, ends up finding out that it was due to the numerous glass blowing facilities in the area that were outputting chemicals into the air. And that's why I was talking about their good air quality monitors, because some species are super sensitive to air pollutants, so you're not going to find them at all. And then there are some that actually like those pollutants, like those nitrophiles. So they're going to want to be in those super eutrophic areas. So you can actually see their crazy maps online of like, of Oregon, of lung cancer and lichen diversity, and it matches perfectly, and it's all just air pollution. Yeah, that's a really cool study. I need to reread that one with the glass blowers.
The mine area that you were to avoid that because of the.
I did. I was worried that it would skew my data. I didn't know if there was like lingering chemical deposition that would, yeah, make it seem like Horse Mountain was just covered in chemicals where it was actually really only that mine. I don't think that there'd be anything lingering. This has been so long, but it's totally possible. So yeah, I just avoided it. It's because it's for efficiency's sake. All of my ground plots were way more difficult to ID. Most of the macro lichens on soil are really, really tiny. And when it's the Forest Service and they're sending a Forest Service crew out, they might not even necessarily be lichenologists. What the FIA crew will do is do a full survey, and then they'll just collect morphospecies that they see, and they get trained on it. But then they're just sending those packets off to real lichenologists who are going to ID them. So if you just can't trust them to collect on the ground as well. Yeah, good question. I would say probably 30% of the diversity was from the ground surveys. And I think that I probably would have had a lot more if I had been able to pick out morphospecies better. Like there's so many cladonia up there, and I guarantee you I missed some just because they look identical, and I thought I'd taken 5 of them and that was plenty, but it turns out no, they're totally separate species. They just are perfectly identical morphologically. Anything else? Okay, thank you guys so much. I know that was a lot of new information.